Hi, today we will be presenting MYC protein stability is negatively regulated by BRD4. This will be presented by Anirudh, myself, Raquel, and Mariah. MYC is a proto-oncogene that regulates proliferation and metabolism. A proto-oncogene is a gene that is involved in normal cell growth, and a mutation in this kind of gene can cause it to become an oncogene, which is a gene that can cause the growth of cancer cells. It's important for MYC to be maintained at homeostatic levels to assure normal cell function. Overexpression of MYC can lead to the cancerous cells, and MYC stability is driven by phosphorylation. MYC has two phosphorylation sites, threonine 58, which causes degradation of MYC, and phosphorylation at serine 62 is responsible for stability and functional MYC. The next protein that's important for this paper is bromodomain protein 4, which is a transcriptional regulator that activates proto-oncogenes. BRD4 regulates transcription in many cancer cell types and has intrinsic histone acetyltransferase and kinase activities, located at its C-terminal and N-terminal ends respectively. It also regulates transcription indirectly through recruitment and phosphorylation of the transcription elongation factor PTEFB. Previous research showed that BRD4 contributes to reactivation of MYC transcription at the end of mitosis and contributes to maintaining cell proliferation and growth. Other reports suggested that BRD4 co-immunoprecipitated with the MYC protein. The purpose of this paper: the researchers are trying to determine what role does BRD4 play when interacting with MYC. Their hypothesis: if BRD4 regulates MYC levels, then we would see a decrease in homeostatic levels.
Other techniques that were used were HAT assay, which HAT assay is a histone acetyltransferase, which this is an enzyme that plays a role in regulating transcriptional genes. And this enzyme acetylates conserved lysine amino acids found on the histone protein by transferring an acetyl group from acetyl-CoA to form acetyllysine. And this modification neutralizes the positive charge of lysine and thus interrupt the interaction between DNA and histone tails. In this diagram, I kind of give you a brief overview of what a HAT assay looks like. So you can see the acetyl group being added and the nucleosome kind of opening up for transcription genes to be regulated and to be expressed. And the reason why it was used in this experiment was to determine what effect HAT activity has on MYC, since we already know that BRD4 degrades MYC, and researchers also wanted to figure out what happens to MYC when HAT activity is present. And we will later observe this in figure 5. Another technique is also nucleosome eviction assay. And in this diagram, I also just showed you what a normal nucleosome assembly looks like. So the black lines are just the DNA, and then the spheres are going to be the histones. And then on the nucleosome eviction diagram, you see that the histones are no longer binded to DNA. So this is just to show the removal of nucleosomes from the binding to DNA. And open the chromatin structure and see if there will be any expression of H3K122 acetylation in the presence of MYC.
So the next technique we're going to be looking at is a CHX treatment. So cyclohexamide is a fungicide that's produced by Streptomyces. We can see its structure right there on the right. So CHX interferes with translocation. Essentially, it interferes with the process of the tRNA molecules and the mRNA molecules binding inside the ribosome, and it blocks the translation process entirely. It's a very useful tool in inhibiting protein synthesis. So you don't get any new gain of protein. You can watch the degradation of one protein itself. And that's what the authors do here, is they apply the CHX through a time course experiment and then followed up with a Western blot to see the half-life and presence of a protein. Later, we're going to see the authors use this experiment to view the degradation of BRD4 and MYC and our MYC P358, P358. Okay, and the next one is the Chase pulse assay. So we look at selected cells that are exposed to a labeled compound. It's a pulse, and then added to a system to be studied. Then this labeled cell or group of cells goes some kind of metabolic process. Generally, it's protein synthesis for Chase pulse assays, but we're going to see what the authors do a little later, and can be quantified through a synthesis of some kind of product. We use this technique a lot just to determine the activity of a cell over some period of time, and it's generally used to study activity of kinases. So here we see a diagram of a typical pulse chase assay. They are usually time course experiments, and so you have a pulse period of 45 seconds in this example, in which you're radio labeling amino acids or proteins of some kind, generally amino acids in protein studies. And then you have an incorporation period of incorporating now your labeled pulses into some kind of medium. And then you do a chase assay, which is just letting it run through its medium, letting the pulses run through the medium for some amount of time. And then quantifying at the end the finished labeled protein or finished labeled product. In this case, our authors use a pulse chase assay to quantify MYC in certain treatments of DMSO, that'll be used as a control, and MZ1, which we want to look at to see how MZ1 affects the half-life of MYC.
An important technique that they use in this paper is the kinase assay. A kinase is an enzyme that aids in the transfer of a phosphate from ATP to another specific molecule, and a kinase assay will measure the kinase activity. They used a few different types of kinases in this paper. ERK1 and GSK3 beta are kinases that are known to phosphorylate MYC. The BRD4 delta kinase is a kinase deficient mutant of BRD4. And BRD4 itself is the kinase that is suspected to phosphorylate MYC. And they also talk about RMYC, or purified recombinant MYC. The steps of this assay were to take the purified RMYC kinase and the reaction buffer, and then incubate that with ATP and allow the reaction to occur. Then the proteins are resolved on SDS page, and they are imaged by autoradiograph, and then they are immunoblotted with the MYC antibody. Another important assay was the proximity ligation assay, and this detects protein-protein interactions with high specificity, and the protein targets can be detected and localized with single molecule resolution, and interactions are revealed in situ, meaning that they occur where they normally take place. And in this assay, it begins with the primary antibody. For this paper, that primary antibody is MYC. And then you have a secondary antibody, which is the PLA probe. And the PLA probe will bind to the primary antibodies. And each of this probe has short sequence specific DNA strands attached to it. And when the proteins of interest are in proximity or part of a complex, these DNA strands will participate in rolling circle DNA synthesis, which you see the start of it in figure C. And then there will be amplification of the DNA circle in figure D. And the fluorescent labeled complementary oligonucleotide probes will bind to the amplified DNA. And this result can be observed with a fluorescence microscope. In figure 1, BRD4 MYC interact in the nucleus. So they wanted to ask, do BRD4 and MYC form a complex? To do this, they used co-immunoprecipitation. Nuclei were extracted from HeLa cells, and they had magnetic beads coated with anti-BRD4 antibody and added that with the HeLa nuclear extract. And then they immunoblotted with anti-MYC. Their positive control was their input lane, and their negative control was the Ig antibody. The result was that immunoprecipitation of BRD4 from HeLa nuclear extracts co-immunoprecipitated with MYC, which you can see in the third lane by the presence of the band in the anti-MYC lane. They concluded that they do coexist in a single complex. Next, they ask, what interaction do MYC and BRD4 have in situ? So they used the proximity ligation assays. The PLAs were combined with the anti-BRD4 and anti-MYC on fixed HeLa cells. They also did this DAPI staining, which shows all the cells that are there, and they observed with fluorescence microscope. Their positive control was the DAPI stained cells, and their negative control was the PLA plus anti-BRD4 plus the anti-nucleolin. They saw that there was robust nuclear colocalization between BRD4 and MYC, which can be observed in the cells that are colored red. They concluded that endogenous BRD4 and MYC coexist in a complex in situ. Is the interaction between BRD4 and MYC direct? For this, they used a reciprocal in vitro pull-down assay to measure the protein-protein interaction. On the left, they have the recombinant BRD4 and recombinant MYC immobilized on the anti-MYC agarose beads. On the right, they did the opposite. So they had the recombinant MYC plus the recombinant BRD4 immobilized on flag beads. Here, the positive control is MYC and BRD4 input, and the negative control is the beads with or without the prey protein. They saw that BRD4 was pulled down by MYC bound to anti-MYC agarose beads. Similarly, MYC was pulled down by flag BRD4 immobilized on flag beads. Here they can conclude that BRD4 and MYC directly interact. How can the interaction between BRD4 and MYC be characterized? So they have this mapped region of BRD4 that interacts with MYC. It consists of the two subdomains BD1 and BD2, and also the hat domain. So then they used the series of recombinant BRD4 mutant proteins in a pull-down assay with purified MYC. So the different mutants were the delta B1, B2, which lack both of those bromo domains, the delta N mutant, which lacks the entire N-terminal half of BRD4, and delta C, lacking the C-terminal region BRD4 spanning its hat domain, but retaining the last 30 amino acids through which the BRD4 interacts with PTFB transcription elongation factor. And their positive control was the anti-His BRD4. Their negative control was beads with or without the MYC. And those are shown in the first few lanes. What they saw here was that the mutants of B1, B2, and the N-terminal were unaffected by their ability to pull down MYC, as you can see the presence of a band. However, delta C failed to bind with MYC, which you see in the absence of the band in the second to last lane. They concluded that MYC binds to BRD4 C terminus independent of either its hat domain or the carboxy terminal segment. What is the BRD4 interaction segment of MYC? For this, they used a series of N-terminal and C-terminal truncations that are shown in the above part of the figure, and then they immunoblotted. So the positive control was the MYC immunoblot, and then the negative control was the beads with or without BRD4. They saw that the truncated mutants that were deleted either in DNA max binding region or spanning the N-terminus were able to pull down BRD4, and a fragment extending from 1 to 63 amino acids did not pull down BRD4. So if we go back here on the figure on the left, you can see that there are bands for all of them except for that region from 1 to 63 amino acids. So they wanted to further define, so they found that BRD4 bound to both 63 to 163 and 144 to 270, meaning that the binding must be dependent on this 19 amino acid overlap. So if we go back here, you can see in their second immunoblot that there was binding for all of them. They concluded that BRD4 and MYC directly interact through BRD4's C terminus between 823 and 1044 amino acids, and the MYC transactivation domain between 144 and 163 amino acids. Next, we move on to figure 2. BRD4 phosphorylates MYC at the threonine 58. So if you remember, threonine 58 was one of those phosphorylation sites of MYC. So to do this, they did a kinase assay followed by autoradiography and immunoblot. And for this, they used ERK1 and GSK3 beta, which are kinases known to phosphorylate MYC, and they also used the BRD4 mutant. And their positive control were these known kinases, and the negative control was MYC. These were the results. BRD4 phosphorylates MYC. MYC did not affect the BRD4 kinase activity. The kinase deficient mutant bound to MYC efficiently, but it was not able to phosphorylate it. MYC phosphorylation by BRD4 was as great as those of the known kinases to phosphorylate MYC. So they concluded that BRD4 is also a novel MYC kinase. In 2B, they ask what residues are phosphorylated by BRD4. And they used a mass spec analysis of MYC phosphorylated in vitro by BRD4. They used their positive control of ERK1 with a known target of serine 62, and their negative control was to use no enzyme. They identified that threonine 58 was the preferred site of phosphorylation by BRD4, and the serine 62 phosphorylation was detected but at lower levels, and the ERK1 preferentially phosphorylated MYC as serine 62 with lower levels at the threonine 58, which was an expected result. And they concluded that threonine 58 was the preferred site of phosphorylation by BRD4. In 2C, they validated BRD4 phosphorylation of MYC at threonine 58. This time they did an immunoblot with MYC threonine 58 specific antibody. They detected that phosphorylation at threonine 58 by BRD4 happened, but not by the mutant. They concluded that BRD4 phosphorylates MYC at threonine 58. And then here the positive control was to use the MYC, and the negative control was the deficient kinase. In 2D, they wanted to confirm these results in actual cells. So the threonine 58 levels were compared between the HeLa cells transfected with BRD4 or with a vector control, and then they immunoblotted the whole cell extract made from these cells. Their result was that the endogenous MYC threonine 58 levels significantly increased in cells overexpressing BRD4, and there were similar results for other cell types. And they concluded that BRD4 phosphorylates threonine 58 in multiple cell types. Here they used a positive control of tubulin and the negative control, which was the empty vector. They asked, does MYC threonine 58 specifically recognize exogenous MYC phosphorylation by BRD4? This time they used the immunoblot of whole cell extract from transfected HeLa cells, and they found that no phosphorylated threonine 58 was detected in samples transfected with BRD4 and a MYC threonine 58A mutant, which you can see in the very last lane where there is no band, and also in the threonine 58 MYC mutant, there's also no lane there as well. And they concluded that the MYC threonine 58 antibody specifically recognizes exogenous MYC phosphorylation by BRD4. Here they also used the tubulin positive control, and their negative control was that threonine 58 MYC mutant. Does BRD4 of endogenous MYC threonine 59 depend on GSK beta? If you recall, the GSK beta was one of those known kinases that phosphorylates MYC. So they did immunoblot of whole cell extract from the transfected HeLa cells with BRD4 or with a vector, and then they treated either with DMSO or with this GSK beta inhibitor. They found that the inhibitor did not inhibit in cellular BRD4 phosphorylation of threonine 58. This concluded that threonine 58 is a major target site of BRD4 kinase activity. And the positive control was tubulin, negative control was the empty vector. In 2G is BRD4 phosphorylation of exogenous MYC-threonine-58 independent of MYC-serine-62. Here they co-transfected BRD4 with MYC or MYC-serine-62 point mutation, and then they did an immunoblot. They saw that equivalent increase in phosphorylation of threonine-58 and the serine-62 levels of the wild type MYC did not increase when they were co-transfected with BRD4. They used a positive control tubulin and negative control was the empty vector. And they concluded that BRD4 phosphorylation of threonine-58 did not depend on the phosphorylation status of serine-62. And BRD4 does not phosphorylate MYC-serine or MYC-serine-62 in cells. For 2H, does differential localization of MYC mutants affect BRD4's ability to bind MYC and thus phosphorylate it? Here they used co-immunoprecipitation of BRD4 and the threonine-58 and serine-62 mutants from transfected HeLa cells. They had a positive control of input and the negative control were the IgBs. Here they saw that BRD4 efficiently co-immunoprecipitated with both of the mutants from the transfected HeLa cells, and threonine-58 was phosphorylated in both wild type and the serine-62 mutant, but not the threonine-58 mutant. From here, they concluded that BRD4 phosphorylates MYC threonine-58 independently of serine-62 phosphorylation, and that BRD4 robustly and preferentially phosphorylates MYC threonine-58 in vitro and in cells independent of serine-62.
All right, so figure three. Figure three is looking at the endogenous BRD4 kinase controls. The figure three is looking at endogenous BRD4 kinase that's controlling endogenous MYC P358, P3-58 phosphorylation and ubiquination. So we look at 3A first. For this figure, we see that the authors use MEFs, mice embryonic fibroblast cells, to make sure that we get an in vivo look, and that MYC phosphorylation is tied to BRD4 in a physiological setting. So previous experiments we looked at earlier were not done in cells, or they weren't done in these MEF cells. So figure A, we could clearly see that we do a co-IP using specific antibodies for MYC, MYC P3-58, BRD4, and tubulin. Tubulin is generally used as a reference marker, making sure that our antibody staining worked essentially, and that our Western blot looks good. So we have a control. It's just wild type essentially. There's a functional BRD4. There's and everything's normal with it, and then we see a BRD4 deleted. We see that when we delete BRD4, we lose MYC P3-58. We lose that domain, but we still see like a increase of the antibodies for MYC staining, like relatively goes up a little bit. And then we don't see any BRD4 at all, which shows that in the presence of BRD4 deleted, we lose MYC P3-58, but actually gain overall MYC protein levels. Then we move on to 3B. 3B is just in situ immunofluorescence using antibodies that are specific for MYC and specific for MYC P3-58. So when you compare the BRD4 deleted of the top two panels versus the bottom two panels, we're looking at BRD4 deleted for MYC and then BRD4 deleted for MYC P3-58, and we see a dramatic difference in these two. We don't see DAPI is generally used as a control and then Cree-GFP is the marker. Um, that so they could do the overlay. But the important panels we see is the merge there on the right, and then the merge, and then the bottom panel of the BRD4 deleted bottom left for MIC and MIC P3 and 58, which you see a significant reduction. We see an increase in the BRD4 deleted for MIC protein overall, actually, but not the MIC P3 and 58, just so we get a visual representation of what we were talking about earlier. 3C now wants to look at, okay, well, we looked at the deletion of BRD4. What happens when we increase BRD4 or we have an overexpression of BRD4? So it's the same as figure A as far as doing the co-IP with antibodies, but now we are testing a control versus an overexpression of BRD4. And so once again, we look at tubulin as a relative level gauge for our Western blot, and then we look at the difference between MIC P3 levels and then MIC and BRD4 between the control and then the BRD4 overexpression. So we have essentially a negative control, as we want. If we overexpress BRD4, we'd see on the Western blot that we'd see on the blot, sorry, that BRD4 levels have increased. That band is larger than the control for sure. But we curiously see a significant increase in MIC P3 58, and then we see a slight decrease in MIC itself. So it shows that there's an overexpression of BRD4, increased P3 and 58, but we see a slight decrease in MIC. Just skip one. Okay. So then we move on to 3D, because we want to look at BRD4 kinase in itself, and we want to see the half-life of MIC now. So we refer back to the technique we talked about in the beginning, or towards the beginning of the CHX assay, which is essentially always done in a time course experiment. Along Chase pulse assay is also done in a time course experiment. But in this case, they do a CHX assay following MZ1. MZ1 PROTAC is a PROTAC, and that degrades BRD4 actually. And so we want to see what the effect BRD4 has on MIC half-lives. So we see an untreated right there for control DMSO. So untreated is just the CHX at the very beginning of the experiment. There's no CHX added to it. And then we see 0, 30, 60, 90, and 120 minutes of a time course. In this case, we see that MZ1 actually loses BRD4 expression. You can clearly see it very quickly loses it. And once again, tubulin is used as a reference to make sure like everything's working. And then we also look at JQI. JQI is known to not have any effect on MIC half-life. JQI is a small molecule inhibitor that binds to BRD4 homodomains and prevents BRD4 binding to chromatin and prevents stabilizing. So JQ1 is used also for a reason to visualize BRD4 binding itself. But by that binding shouldn't have any effect on MIC half-life. So they prove that through this time course of the CHX assay. And we can see that MIC itself through the MZ1 has a prolonged half-life, prolonged MIC protein levels. We see JQ1 is relatively similar to a control, which gives the conclusion that MIC stability is unaffected by JQ1, and that makes sense because JQI1 is yeah, JQ1 is tied to BRD4 binding, and MZ1 is tied to BRD4 degradation, and MZ4 extend the half-life of MYC. So the presence of BRD4 correlates with P3 58 levels and MYC stability. 3E, BRD4 correlates with P3 38 levels, 58 levels, sorry, and MYC stability. And so similar experiment to 3D actually, except they use a slightly different approach using a pulse chase assay, visualizing the half-life of each of the proteins. With MYC immunoprecipitated with HeLa cells, and then they use DMSO and MZ1 as they're observed here. So they pulse for 2 hours, and then they chase for the 90 minutes. The pulse here is sorry, they pulse for 20 minutes. Sorry. They use a methionine cysteine tag on 35 serine, which attaches to that MYC protein level. And then they were immunoblotted. And so looking at the graph, the top panel E right there is the results of the chase assay itself, where we see DMSO is our regular control, and we see the degradation of that protein, the protein level over 90-minute period. And then we see the MZ1, and we see that it degrades a lot slower, and we see more protein, which shows that the anti-MYC, that shows that MYC P3 in the presence of MZ1 is non-existent, which makes sense because MZ1 degrades BRD4, which also shows that makes sense because BRD4, the antibody for BRD4 doesn't pick up anything at all because BRD4 has been degraded by MZ1. And we actually see a slight increase in MYC protein levels throughout this time. And once again, tubulin is used as a reference, making sure that tubulin is still transcribed and present. So it shows that the deletion of BRD4 stabilized the MYC half-life. These two are correlated. BRD4 deleted or degraded through MZ1 has increased MYC half-life. Moving on to 4A, then we look at BRD4 phosphorylation into exogenous MYC threonine 58, resulting in protein degradation. So once again, we go back here and we see that MYC threonine 58 has disappeared now, degraded through the loss of BRD4. So now we want to take a closer look at that. So in this case, they use HeLa cells co-transfected with MYC and either wild-type BRD4 kinase mutant, or an empty vector. So now they're looking at particularly the BRD4 not functioning with a kinase mutant and not through MZ1. So they're not going to use MZ1 to degrade it anymore. They're just going to look at a defective BRD4 kinase mutant. And then we can see that at the top top gel there, when the anti-ubiquitin staining, the antibody for ubiquitin, we see that we lose that MYC BRD4 kinase. We lose that second 60 kilodalton band because there's nothing being tagged for degradation there without the BRD4 kinase mutant. So that MYC isn't being tagged for degradation. But we do see it for the MYC and regular functional BRD4. That's that second band right there. Oh, sorry guys. Oh, what happened? Yeah. And then the second gel right there is just showing the presence of MYC throughout these. And then the bottom is showing the presence of MYC P3 58. That's the important one. Because we see that now with a dysfunctional BRD4 kinase in that last last band, last lane, sorry, last lane, we don't see any more MYC P3 and 58. We still see MYC and we still see the antibody for BRD4. That makes sense because, or sorry, not the antibody. We still see antibody staining for BRD4, which makes sense because it's a defective BRD4 kinase mutant. It's still going to show up, but now it can't do its job at the phosphorylation level of MYC P358. And so that next they look at a protein, they want to look at the protein stability, right? And so they perform a CHX assay for 16 hours incubated for a 3-hour period. They're particularly looking at protein levels for MYC and the MYC BRD4 kinase and the MYC regular. And so we can see that the MYC plus the BRD4 wild type, we do see a degradation in MYC right there at the 90, 120, 180 level, pretty consistent degradation. But with the BRD4 delta kinase mutant right there, we don't see a drastic reduction in the degradation of MYC. It's just showing that it's no longer phosphorylating at the P3 58 area, and therefore leads to better MYC stability. So MYC phosphorylation and stability depend on functional BRD4 kinase activity. And the final figure is looking at that promoter activity for regulating the P3 58 and MYC stability itself. So they use a luciferase assay. There's a CDK4 promoter for MYC that's modulated by BRD4 kinase activity. And so the authors create this CDK4 E-box luciferase reporter plasmid. And then they have that plasmid just as a control, and then they co-transfect it with other factors, such as just MYC, MYC BRD4, MYC BRD4 delta kinase mutant, BRD4 regular, and just the BRD4 delta kinase, just to see the expression levels. And so when you do this assay, you see the control, the relative control right there as a reference point. And then you see MYC. We see that there's a relatively large promoter activity when there's just MYC being expressed. But then we see MYC plus BRD4, we see a lesser amount of promoter activity, which makes sense because then you have the BRD4 phosphorylating in that P3 58 region right there, which leads to, MYC degradation and less MYC in general. And then we see the MYC plus BRD4 delta kinase. It shows that, well, this is not significant at all from MYC. It's pretty similar, which shows that now this BRD4 delta kinase has kind of destroyed the function on degradation of MYC that it generally has. And then they use BRD4 and then BRD4 delta kinase as kind of controls. They're like negative controls. We expect that we can expect that we see promoter activity for BRD4 and BRD4 delta kinase when we're just expressing these two. And so yeah, in this case, they're able to say MYC stability and function are regulated by the BRD4 delta kinase, particularly in BRD4.
Now for figure 5, you will see for this title is going to be MYC inhibits BRD4 histone acylation and nucleosome disassembly, which is also known as nucleosome eviction. So in figure A, in this blue box, the question that's being asked is, can HAT still acetylate histones in the presence of MYC? And the control is going to just be the BRD4, because we see that there is some expression of H3 and H4. And then we also see as BRD4 and MYC are added together, as MYC is increased, we do see BRD acetylation lessening. So the conclusion that the researchers did find based on this figure alone was that MYC does in fact inhibit BRD hat activity. And then for the next figure, figure 5D, this is another important figure because this one focuses on the nucleosome eviction. So this is the method that was used. And this is just a question that's being asked is, does MYC affect BRD4 nucleosome disassembly? Again, nucleosome eviction, in other words. And based on these results, you could see that acetylation of H3K122 of BRD released the free DNA from the nucleosome. So you see the free DNA all the way at the bottom. And then as MYC is added, increase of the ratio from BRD4 and MYC is increased, you see then it was inhibited. The acetylation was in fact inhibited. So based on the results from this immunoblot, we see that MYC binding to BRD4 does inhibit its ability to dissociate nucleosomes. Another important figure from figure 5 is going to be figure 5F. And this question is asking, does phosphorylation of MYC affect its ability to inhibit BRD4 hat phosphorylation or hat activity, sorry. And this was also because this was wanting to be figured out just because we wanted to see if the phosphorylation has any effect on MYC's ability to inhibit hat activity. And based on the results, we did find that unphosphorylated MYC does reduce BRD4 acetylation of histone H3, but phosphorylated MYC has no effect. So this shows that inhibition of BRD4 hat activity is dependent on phosphorylated MYC, because we do see that it's only when it is phosphorylated, there is no effect of inhibiting the hat activity. Next, we'll move on to figure 6, which figure 6 is an important figure as well because it does show the ERK1, how it interacts with BRD4 and how it inhibits its BRD4's kinase activity. So in figure 6A through 6C, we see the question that's being asked is, does ERK1 and BRD coexist? So this was determined to see if they are working together, that to work with MYC, and the methods that's being used is the co-immunoprecipitation and pull-down assay. And the results here, they do show that there is interaction between BRD1 and ERK1, and you can also see that in 6B, where you do see the anti-ERK1 and the anti-BRD4, and then when you have them together, you do see that they are coexisting together. And also in A and C, you also see the same thing, just a different way of showing the data. And the conclusion was based, researchers did determine that BRD4 and ERK1 do coexist together. And to further expand on that, the researchers also wanted to see if ERK1 is where it is located on BRD4, and if they compete together with binding with MYC. So method in this one is just a pull down assay, and the results in this one does show that MYC is binding to the N-terminus, and that is because you see in D, when the N-terminus is deleted, there is no ERK1 being pulled down. And then when MYC is when the C-terminus is being deleted in MYC in figure G, we see that there is no pull down of MYC as well. So then this determined that ERK1 binds to the N-terminus and the MYC binds to the C-terminus. So in conclusion, it was determined that BRD4, MYC, and ERK1 form a complex, and so that they work together. Finally, also another important part of figure 6 was figure 6I, and this question was to figure out, does ERK1 inhibit BRD4 kinase activity? And obviously method used is going to be kinase assay. And this was, based on the results, you could see that MYC was only phosphorylated when ERK1 was phosphorylated as well. And it uses the TAF7, which is it is also just a substrate that it does not get phosphorylated from ERK1, and we are able to observe that ERK1 inhibits BRD kinase activity based on looking at the first 3 columns. We see that BRD4 plus MYC, there is phosphorylation of each, and then as BRD4 plus the complex that BRD4, ERK1, and MYC make, there's also phosphorylation, but in order to compare that, we did use the substrate of TAF7 to show that ERK1 inhibits BRD kinase activity directly.
So our conclusions are that BRD4 regulates MYC and maintains the homeostatic levels of the MYC protein. So we see that BRD4 directly stabilizes MYC protein at the phosphate, the p-threonine 58 phosphorylation site that leads to its ubiquination and consequential degradation to keep homeostatic levels of MYC protein. JQ1 was found to reduce MYC transcription, but has no effect on the MYC protein stability itself. And MZ1 degrades BRD4 that decreases the MYC transcription, but increases its protein stability. And here's our bibliography. Thanks for listening.